Cancer screening guidelines can be pretty confusing for both the patient and for the physician. So we want to go back and cover some of the concepts of basic cancer biology that we talked about first year to see why the situation is so confusing right now and to see what hope we have for things getting better in the future. This is just to remind you that every cancer is the same. Every cancer arises from some cell in the body that makes this decision incorrectly. It decides that it is the kind of cell that ought to divide when the answer was no, it isn't the kind of cell that ought to divide. So, since every cell in the body has the same machinery for making that decision, if this decision is made incorrectly in a heritable way, every cell can turn into a cancer. So at that level, every cancer is the same. At the same time, every cancer is different. And the reason that they're all different is they're all gathering and interpreting information differently because each tissue type uses information in a slightly different way. So that cell cycle machinery over here that we just talked about is the same in every cell, but all this information gathering capacity, all the signaling that's going on, all the sensing of the environment, that's all different. So each kind of a cell might take a similar kind of a signal and interpret it in a completely different way. For example, a hormonal signal might come to a breast cell and say, this is a good time to grow. A hormonal signal might come to a prostate cell and be interpreted as, go ahead and stay alive. So it's being interpreted, the same kind of a hormonal signal is being interpreted very differently by these two kinds of cells, even though those are both hormonal signals. So all of that signaling comes down to these four things. Should I proliferate or not proliferate? Should I die? Should I not die? In the one case that we just talked about, the signal is a grow or don't grow signal in the breast, but in the prostate, instead, those hormonal signals, are die and don't die signals. And it's the balance between all of those that tell us whether or not we're going to overproliferate or not. Since every tissue is interpreting this information differently, every tissue has a different probability or different set of steps it has to go through in order to become cancerous. So the most important thing I want you to get out of this little video is this slide. I want you to remember this concept because it really is key to understanding why screening guidelines are different. Every cancer is the same because every cancer has to grow too rapidly. Every cancer is different because it's making different decisions, and these are the decisions ultimately that every cell has to make in order to become a cancer. So why is it that screening guidelines are different? Because different tissues have different ones of these steps that they have to overcome. They all have to be made in order to become a cancer, but some cells don't have as much emphasis on any particular one. So for example, if we look at a hematopoietic stem cell and we ask how does it turn into a leukemia, well, it's a stem cell, so it doesn't have to worry about getting limitless replicative potential. It already has all the machinery in place that says you can make telomeres, you can live forever, you can make as many cells as you want, you're a stem cell. It doesn't have to worry about getting sustained angiogenesis because it's living in a place with lots of nutrients and oxygen, so that's not an important step for it. It doesn't, again, have to get self-sufficiency in growth signals because it's a stem cell that's already going to be growing very rapidly. So as you can see, some cells already have many of these boxes checked off. They don't have to worry about overcoming those things. And that's why leukemia is sort of a one-step cancer. All you really need to do is hit the accelerator a little bit harder than normal, make a little bit too much of one of the cyclins, for example, and you can turn a hematopoietic stem cell, get it to differentiate into one of its pathways, and then grow too rapidly, and then you can get leukemia. If we look at other cells in the body, for example, a prostate cell, one of these cells doesn't have any of these things going for it. It doesn't have a good source of blood. It's not supposed to grow very rapidly. So it's going to have to overcome all of these steps. The basic point is that as we look at each kind of cancer, we have to look at the kind of a cell it comes from, and that will tell us how many of these steps have to be overcome in order for it to over-proliferate. And that number and the way it's going to get around each of those blocks is unique to each kind of cell. So if we want to ask why cancer screening guidelines are different, then the answer is going to have something to do with each of the tissues having different ways to get from where they are to being a cancer, and that's going to determine both the probability and the pathway that they have to take in order to get there. Let me illustrate the point with the best example, the easiest example, the colon cancer model. So the cancer screening guidelines for colon cancer are straightforward. Everybody agrees on them. They're easy. Why is that? Because colon cancer itself is straightforward, stereotypical, goes through a standard progression, and that gives us the opportunity to anticipate how it's going to behave, and it's going to behave that way in a large fraction of the cases, so screening is going to be an effective tool. Why is that? So let's start back where the colon carcinoma starts from, over here in the normal epithelium. So there are stem cells sitting in the crypts in the colon, and they're going to give rise to this rapidly proliferating cells. Those are going to move their way out to the edges of the lumen, and then they're going to undergo apoptosis and die off. This process is going to take something like 5 days. So if you want to evade this process and not die in 5 days and become a cancer, the first thing on your mind isn't going to be about getting a blood supply. It's going to be about shutting off that apoptotic signal. So as soon as you differentiate, you start this process that will ultimately end up in apoptosis. So what you need to do is shut off that differentiation signal. The differentiation signal comes through this signaling pathway that goes through APC. So then if we want to become a cancer, the first thing we have to do, shut off the differentiation signal, mutate APC, and give ourselves a little bit of breathing room. Otherwise, we're going to be dead in 5 days. So this is why colon cancers always have either a mutation in APC or one of the things that feeds it, signal to it, because that signal needs to be shut off. That's the very first thing that has to happen. Once that has happened, now we've got a little bit of time. We can start growing a little bit. And now we have these other questions that we need to answer. We need to get over all of those 7 barriers in order to become a cancer. But the first one has to be this APC mutation. Once that has happened, then there's going to be a slow progression through steps that are going to lead us to becoming a frank carcinoma. But those steps take a while, and this is all happening in a relatively accessible place. We can see this happening, we can go in and look, and we can see when something like this is formed. So we know that a problem is occurring. The process takes about 10 years, and it's about the same in every colon carcinoma. So a nice stereotypical progression means predictable behavior. We can tell exactly what's going to happen next, and we know how long it's going to take. That's why screening is such an effective tool in this particular case. This brings up the point that since lots of cells start off on this pathway towards cancer, they start to proliferate a little bit, but they don't necessarily make it. That means that we're going to find things that look like they're growing rapidly, but they may not be dangerous. So screening is always going to come up with a lot of things that are doing something abnormal, but we have to be able to tell the difference between when abnormal is dangerous and when abnormal is benign, and we would be better off just letting the process go on instead of intervening. So if we're looking at the things we find during a screen, we look down here at screen 1, we're going to hit some things that are growing rapidly. These ones are all benign. They aren't going to harm anybody. They'll go on growing a little bit faster than normal, but they aren't ever going to solve the problem of how to become a frank cancer. The guy over here in red is a dangerous one. He hasn't happened yet during our first screen, and that guy's not going to make it to the second screen because this is a dangerous event. and so our screening interval was inappropriate for finding this particular kind of a cancer. So we might find one over here early on that was dangerous, but we're going to find along with it in this screen too all these other events that are benign. So it's going to be very important for us to be able to tell the difference between things that are dangerous and things that aren't dangerous, because we're going to find both when we do screens. And that's where the promise of technologies like microarray analysis are supposed to help us, and are helping us a lot in some cases, but not so much in others. So to remind you, every cell in the body has something like 20,000 genes that it can express. It's only expressing a subset of those, and expression is at least partly through transcription of genes into RNA molecules. Of course, much more than that has to happen before a gene gets expressed, but it's easy for us to measure lots of RNA molecules, so we're using that as a stand-in for expression. If we look at a normal stem cell, over here, it's going to be expressing some of its genes a lot and other things less. The things that are important for it to be a stem cell will be expressed a lot, but as it differentiates, those things will be expressed at a lower level. It's not making cell types specific messages very much. As it differentiates, those will go to a higher level. So this is just sort of the general principle. We can learn something about what a cell is thinking by collecting all of the RNA molecules in it and seeing how many of different genes, how many transcripts are present from different genes. So if we do that, what's gone on with a cancer cell is it has gone from some normal profile back to a more stem-like profile, so it's re-expressed some of the stemness genes, and it may be overexpressing or underexpressing some other more tissue-specific genes. But the point is, it's got increased expression of things that might be oncogenes, they might be driving the tumor, and it's got decreased expression of things that might be tumor suppressors that also might be driving the tumor. So we should be able to look at the profile of RNA molecules and learn something about what that particular kind of cancer is thinking, what that particular cell is thinking. And that will tell us something about what we should do next. Is this a dangerous profile, or is this the profile of a cell that hasn't yet figured out how to be dangerous? So then we take all that information from the RNA molecules, and we take tumors from different people, and we say, okay, here's a bunch of dangerous tumors. What kinds of RNA profiles do we find for different genes in those tumors? And we can line them up like this, and we can find that there are clusters. Here's a bunch of tumors that were all expressing this set of genes at an elevated level. A bunch of other tumors, these ones don't have elevated expression of those genes. And this just underscores the point that I made earlier, that every cancer is different. They've all solved this problem of how to get by the 7 deadly sins in different ways. But there aren't an infinite number of ways to do this. So when we look at different tumor samples, we're going to find sort of strategies that were successful, and there will be a finite number of them. In the case of something like breast cancer, we got lucky, and there were only 3 general categories of ways that cells became breast cancers. So now we can cluster them together, recognize that that's true. We can take any given patient and say, which group do you fall into? You fall into this category. Well, we've already developed a treatment for that category, and it's effective, so we know now that we can treat you that way. If you don't fall into one of these categories, we can say, well, then this is probably one of the benign ones. It hasn't solved the problem because dangerous tumors have one of these profiles. So in the case of breast cancer, we've done quite well in terms of putting those cells into groups, and then we can study them to see which strategies work, which interventions are effective for treating patients with that kind of a cancer. And so that tells us how we should proceed clinically. So we can learn something about what tissues are thinking by collecting their RNA molecules and doing this kind of analysis, and that's been fairly effective for analyzing for both diagnosing and treating breast cancer. situation with something like prostate cancer has not been quite so successful. So we've tried the same strategies. We take different specimens from benign and from invasive prostate cancers, and we measure which RNA molecules they're making, but they don't cluster into categories quite as well. Probably the natural history of a prostate cell says there are lots of ways to solve this problem. It's a type of tissue that doesn't grow very rapidly, so it's going to find all sorts of creative ways to overcome the 7 deadly sins, and it may not necessarily cluster into groups of things that we can study independently. So some cancers have been quite amenable to screening because they have a natural history where they are very stereotypical in their progression. They have to pick up mutations in a specific order, and they have to go through specific processes. They're in places that we can see. Other cancers have been much more difficult to study because they're in inaccessible places. They don't cluster into groups of things that we can study as groups. We have to take each one on its own, and we can't tell the difference easily between dangerous and benign versions of hyperproliferation. So the currently active areas of research are to try to study these ones that have been more reluctant to yield their secrets, and hopefully what we will do is get more information and understand what the key screening targets would be for those kinds of cancers, so that we can get better guidelines in the future and do a better job of screening for other kinds of cancers. So that's it. What you should remember is the 7 deadly sins all have to be overcome. Every tissue type has a different number of those to overcome, and it has different strategies for doing it. The more stereotypical the progression of a cancer is, the easier it's going to be for screening. And the more we learn about what a particular cell type is thinking, the better we'll be at determining whether we're looking at a dangerous or benign hyperproliferation.